Hello students, Pranam. Hope you all are doing well. So today let's start up with our topic, and before that we must recapitulate whatever we have discussed in the last class. Last class we have discussed about the plasma membrane, its function. In that we have discussed about diffusion, osmosis and types of solution. Then we have discussed about the behavior of the cell in three different types of solution. These are hypertonic, hypotonic and isotonic. Whenever we are keeping a cell in hypertonic solution, maybe it's a plant cell or animal cell, the cells are gaining water, and when they are gaining water they swell up. So the plant cell never breaks because it is having the cell wall, but the animal cell may burst. Just opposite, when the cells are kept in the hypertonic solution, which is more concentrated than the solution of the cell, there both type of cells they are losing water and they shrink. No. So another type of solution is there, that is called the isotonic, where the solution, the concentration of the solution outside and inside the cell, it is same. That's why the shape of the cell, it is not changing, it is similar, because net flow is here 0. How much it is moving out, that much getting in, means endosmosis and exosmosis, they are similar. So this is about the previous class. Now let's start with a new topic, that is the cell wall. And you all know the cell wall is a identifying feature of the plant cell, which is present only in the plant cell but not in the animal cell, which give extra protection to the plant cell. Got it? And hence it makes the plant cell more rigid and tough. So let's know more about the cell wall, the unique feature of the plant. Cell wall is a rigid, non-living covering present outside the plasma membrane in plant cell. Animal cell lack a cell wall because we need more flexibility. Plant cell is made up of cellulose, which is permeable to water, solutes and gases. In fungi, the cell wall is made up of chitin. So, in case of the plant cell, fungi and bacteria, all are having the cell wall, and in these three cases, cell wall are made up of different materials. Then we will see that the plant cell, it is made up of number of cells, many cells. So how the cells are joined with each other? So the cells are joined with each other by means of a cementing material that is called the middle lamella. So what is the middle lamella made of, or what is the chemical nature of the middle lamella? Here you can see the two adjacent cells, it is packed by the middle lamella that is made up of chemicals called the calcium and pectin. It is not given in your book, but the information is very much necessary, so you must note down that the cells are packed or attached to each other by means of a cementing material called the middle lamella, which is made up of calcium and pectin. It is just like a wall whenever it is made by the bricks. How the bricks are attached with each other, how they are joined with each other. The bricks are joined by the cement? No, the bricks are joined by the cement or with the cement. Like that, the cells are, here you can see just like the cell, they are joined with each other by means of this middle lamella, which is made of calcium and pectin. So whenever the cells are joined with the cementing material, means they are cemented, but still they need to be communicated with each other. So for their communication, some gaps must be there, and through the gaps the materials are moving from one cell to another. So there are special type of gaps or channels are there. They are called the plasmodesmata. As you can see in the diagram, this plasmodesmata, these are the channels. They are connecting the neighboring cell. And through the plasmodesmata, the various types of materials which are required for the cell, they are moving from one cell to another. Not only this, water when moving through the cells, it is also moving through the plasmodesmata and moving from cytoplasm to cytoplasm. So next, let's learn about the various functions of the cell. So what are the functions of the cell wall? It gives definite shape to the cell. It provides mechanical strength to the plant. It protects the cell against mechanical injury and pathogens. It helps in transport of various substances across it. Cell wall helps the plant cell to withstand a lot of variations in the surrounding environment. Cell wall prevents the bursting of cell on endosmosis as it is quite thick and rigid. So whenever due to endosmosis water is entering into the cell, as we have discussed in our recapitulation part, that whenever in a hypotonic medium the cells are there, the cells are gaining water. So when it is a plant cell, it is not bursting. But when it is an animal cell, it is bursting, because animal cell is not having cell wall, so it may burst. But in case of plant cell, as it is having the cell wall, that time it is not bursting, but it is very much turgid. Okay, so it is very much turgid. So we must know what is this turgid and what is the turgor pressure. So turgor pressure or turgidity, it occurs due to the entrance of the water inside the cell. So then the water is exerting a pressure or it is pushing against the cell wall. So when the plasma membrane is pushed towards the cell wall due to that pressure, and that pressure is called the turgor pressure, and in that condition a cell is called as in turgid condition, or it is the turgidity. So why this turgidity condition is there? Due to the inward flow of water, or due to endosmosis, the cell is getting water. That water is creating pressure against the cell wall, and due to that pressure the cell will become turgid. Okay, so this turgidity is very much important for the plant. Due to that the leaves are flattened, and for that easily they are getting the sunlight. So whenever these turgid cells are kept in the hypertonic solution, so that time what happen? They lose water, and the content of the cell, the plasma membrane, the protoplasm, they shrink, they move away from the cell wall, and this process is called plasmolysis. So in plasmolysis, shrinking of the content of the cell is there away from the cell wall. When it occurs? When we are keeping a cell in a hypertonic medium. Got it? So here I want to show you something which is in our day-to-day life we have seen, that is about the touch-me-not plant. You all have seen the touch-me-not plant. So now let's see how the touch-me-not plant, it closes its leaves whenever we are touching it. Got it? Whenever we are touching the leaf or any part of touch-me-not plant, that time we can see the leaves are closed. So how the leaves are closed? And this is only the game of water. So how? Let's know. So here the scientific name of this touch-me-not plant is Mimosa pudica, and whenever we are touching the touch-me-not plant, some chemicals are secreted, and due to secretion of the chemicals, the cells are stimulated, and hence the whatever amount of cells the cells are having the water, all the water withdrawn from the cell and deposited in the stalk of the leaves. Okay, so what here we have discussed, that whenever we are touching the touch me not plant, that time chemicals are secreted and they are stimulated. The which process? Exosmosis process. It means the water, it is withdrawn from the leaves and deposited in the stalk of the leaf. Now the cells are losing the turgidity, means now they are plasmolized, or you can say they are shrinked. So due to shrinking of the content of the cell or due to loss of turgidity, now the leaves are closed. So here you can say how the turgor pressure and turgid condition is very much important for the leaves, because if the leaves are not turgid, then they will not be getting the sunlight in a proper way. So with this, here we come to the end of the today's topic, and you must go through the lines of NCERT book. Okay, thank you.